Okay. Today we are going to keep rolling, talking about transport, which we broke into at the end of class on Wednesday. So we've gone over what membranes look like. You reviewed that on your own. We talked a lot about electrochemical driving forces by identifying chemical gradients and therefore chemical forces, electrical attraction creating the electrical driving force, and then how we combine them together. So now that we know which direction a molecule wants to go relative to the intracellular fluids versus the extracellular fluids, we want to know, can it get there and how, okay? So our first division is between passive transport and active transport. Passive transport being movement that's not going to require any additional energy, right? So if you've identified the chemical driving force, the electrical driving force, and your overall electrochemical driving force, sorry, I'm distracted by people in the hallway, then you already know what direction passive transport would be, right? So passive transport is going to be in the desirable direction. So if you have an ion, its electrochemical driving force is into the cell. Passive transport would be any movement into the cell. If you have something like glucose or oxygen, you don't have to pay attention to any electrical stuff because they don't have electrical charges on them. So you can just find the chemical gradient, chemical driving force, whatever direction that goes, that's the direction of passive transport. So we think about this as downhill movement, down the gradient, when we typically come up with our mental image for it. We're going high to low. There are a couple different types. The types are going to depend on properties of the molecule you're looking at and properties of the membrane. So we're going to see that our types of passive transport, simple diffusion. We're going to have stuff that can just directly go across the membrane on its own. For facilitated diffusion and diffusion through channels, we're going to need help. We're going to need to involve a membrane protein that's going to help whatever we're looking at get across our membrane. Okay. So for simple diffusion, we don't need any membrane proteins. Phospholipids, just that basic membrane bilayer, fine for us. Okay. This is because simple diffusion, when we talk about it, we're thinking about, usually we're really thinking about oxygen and carbon dioxide, but if we want to think more broadly, we're thinking about simple non-polar gases, small things that are able to cross the membrane on their own. Okay. So simple diffusion, we're just able to kind of float through the membrane like a ghost. Okay. So this means that we're going to have fewer factors influencing the rate of simple diffusion than our next couple types. Okay. So for simple diffusion, we need to know the magnitude of the driving force. So basically, if there's more of a gradient, that would be more of a chemical driving force, a larger one, greater push, right? You fall down the hill faster, that would influence simple diffusion, it would happen faster, it would influence the rate. Little trickier, the membrane surface area, right? So if we're thinking a cell has more surface area, it's bigger, more likely that an oxygen molecule is going to bump into a bigger cell. So if we have greater surface area on a cell, right? So more membrane for us to bump into, this increases the rate of simple diffusion as well. And finally, membrane permeability can also have an effect on this rate of diffusion. So if it's just easier to get across that membrane in general, then we'll influence the rate of diffusion basically by speeding it up. Okay. So we're looking at rates of transport, which we're starting with simple diffusion. Really, what we're thinking about mainly on the y-axis, let's start with the y-axis, right? We're interested in rate of transport. So we're interested in how much is moving, right? So net flux, net is our overall flux. We're thinking flow. So the overall flow of whatever it is we're tracking. If it's something that's undergoing simple diffusion, usually we're just gonna see as we increase our concentration gradient. So if there's more of a difference between the extracellular fluid and the intercellular fluid, we're gonna have more movement. So that's that first piece influencing the rate of transport, that magnitude of the driving force. That's kind of our energy for this process. Places where we'll see like membrane surface areas playing a role can be in things like capillaries and in the lungs, or even in red blood cells. We're going to see some specialized cell types who try to make it so that they have a lot of surface area, so they have a lot of contact with things that undergo simple diffusion, by which usually in physiology we mean carbon dioxide and oxygen. Right? So lots of surface area means you've got lots of places to touch air. So there's lots of ways for air to get in. These are like potential mental abbreviation. So when we think about membrane permeability, a couple things that go into that. So different diffusing substances could, in theory, have different membrane permeability because they have different lipid solubility, right? So what's influencing whether something can do simple diffusion at all, right? Like if it's nonpolar and small enough, they can like sneak through those phospholipids and sneak through particularly that thick layer where we have those fatty acid tails facing each other. So we've got this nonpolar hydrophobic region, right? So some molecules are going to do that easier than others, even if they can both do it. So if you have something that's more lipid soluble, right, that's a lipid that you're going through, so something more lipid soluble would be able to go through more quickly. Something that is lipid soluble, but not quite as lipid soluble, will go through a little more slowly. That also relates a little bit to the size and shape of the diffusing particle. Something that's bigger, more complex in shape, even if it can get across a membrane, might do it a little more slowly, might have a little more difficulty, even if it can do it in the end. So that would also potentially slow down this simple diffusion. Fun one, but not particularly relevant in human physiology, is temperature, right? Not relevant because we have a core body temperature that we're trying to maintain through homeostasis. But the way it works, right, is all your molecules, all your atoms, everything's always just kind of jiggling all the time. Temperature is really just like increased jiggling, kind of, or at least that's how I think about it, right? So when we have higher temperatures, that means things are like a little more unstable, they're a little more active, they're moving a little more. So that would speed up diffusion because it's in general kind of speeding up movement. The final one, the thickness of a membrane. So when we think about membranes and we draw them from our typical like cell level or little basic cell, we just draw a phospholipid bilayer with two phospholipids kind of facing each other, tail the membrane. But in physiology, sometimes when we think about membranes, we're going to have other factors influencing how thick a membrane kind of behaves. So you can think about like, but if you have a cell and it is, you know, still just a phospholipid bilayer, but what if there's like a thick layer of mucus stuck to the outside, right? In essence, that makes the membrane sort of thicker from the perspective of a molecule because it has more space that it has to travel across. If you have more space that you have to travel across, maybe it's a little harder. Increasing the thickness of a membrane is going to slow down simple diffusion of it. That's slower. Okay. So here we have a slightly altered image from the one we saw previously, right? So this is still simple diffusion. We're still looking at net flux on the y-axis, so still how much is moving. Here we still have our concentration gradient as our main kind of driver, right? That difference between the inside and outside of the cell is still mostly what's powering this, right? So as you have a concentration gradient increasing, we're increasing the flux. But here we're noticing that if you have something that's highly permeable, so finds it really easy to get through this membrane for a variety of factors, we're going to have something diffusing more quickly. So this net flux is going to be higher faster. versus if we have low permeability of that membrane. It's going to happen, but it's going to happen a little slower. So here's an example of one of these cases. Is it showing up? Let's see. Did I finally get it? So it'll do it automatically. That would be excellent. Is it letting anybody answer? Is it giving anybody an end? Oh, we got two. Okay, cool. So it's working. Thinking about why it might be the case that when we have fluid in the lungs, oxygen is less able to get into the blood. We're thinking about this idea of simple diffusion. Which of these factors might be related to this? Okay, take another second or so. Take a look. Yeah, perfect. So this is one of the examples of where that membrane thickness calculated. Okay. So water doesn't really change anything about the membrane itself from the perspective of its temperature or the perspective of like how the lipids behave. But if you're an oxygen molecule in the air in the lungs, suddenly you have to get across a layer of water to get into the cells, right? You've, in essence, kind of thickened that membrane from the perspective of the oxygen. So this kind of decreases the capacity of oxygen to get into your blood. So you're going to get less oxygen if you have fluid buildup in your lungs. Now, oxygen can travel through water just like it can travel through membranes. It's small. We'll talk about the solubility of oxygen and carbon dioxide in water when we get to A&P. So that's coming up in the future, but the distant future. These are some questions that are in your book, so you can go through and check those over yourselves. Make sure you understand simple diffusion. Next, we're going to start to think about the pieces of this puzzle that require membrane proteins. We're going to start with facilitated diffusion and carrier proteins. Okay. So when we say that something is a carrier, if we say we have a glucose carrier, we're talking about a membrane protein that goes all the way through the membrane. So this is a transmembrane protein, integral protein, going all the way across. Carrier is going to have a binding site for the molecule that it's carrying, right? So it's going to like physically grab the glucose in the case of a glucose carrier. When it binds to that particle, binds to its molecule, whatever it is carrier for, that creates a shape change. And when that shape change occurs, changes the shape of the protein and has the effect of kind of flipping it across, flicking that molecule to the other side. So binding is going to occur for a carrier protein one side at a time, right? Because you're either in one shape or the other, right? So before we bind in a glucose, we have this kind of pocket for our glucose molecule facing the extracellular fluid, the ECF, outside the cell. Glucose binds in. When glucose binds in, we have all sorts of kind of reactions happening between the R groups of the different amino acids and all the little chains coming off, whatever, right? So the protein changes shape, just like an enzyme kind of changes shape, right? When it changes shape, it's moving that binding site to the inside of the cell, the intracellular fluid here, right? When that binding site is facing the inside of the cell, it can also be facing the outside of the cell, right? It's just one in one place at one time, gets rid of its glucose, and then goes back to its original state. So that's what a carrier protein looks like when we talk about these transporters. Sometimes we'll use the word transporters kind of equivalently. This is what we're really talking about. So binding the molecule into one side of the membrane of into one side of a protein, crossing a membrane at a time, flipping over and then going back. Okay. So when we talk about facilitated diffusion, we're talking about things like glucose and this glucose transporter. Right? For it to be facilitated diffusion, glucose would still be going down its chemical gradient, right? So in this example, for this to be facilitated diffusion, we'd still be talking about a situation where we have high levels of glucose here in the blue region in that extracellular fluid, low levels of glucose in the intracellular fluid, right? But glucose isn't able to cross right through phospholipids, right? It's not small and nonpolar, it's a little bigger, and it's got some OH groups hanging off of it, right? So it can still go across, it can still passively diffuse across, it just has to get the assistance of this protein. So this still wouldn't require energy, and it's still in that kind of favorable direction, moving towards equilibrium. Now, when we think about rate of facilitated diffusion, it is going to look a little bit different than the simple diffusion because we now have the second player in here, right? We've got this carrier protein. Okay. So it is true that each of these proteins, think about them as like, they have to flip to get the glucose in the cell, right? They have to actually change shape. Think about it as like each of them takes a certain amount of time to do that, right? Like it takes a while for them to flip over, right? So each of those carrier proteins is going to have its baseline rate of transport, which is going to put a maximum on our ability to move whatever we're moving, but in the previous example, glucose, right? We're going to put a kind of top line where we're going to plateau because you can't go faster than all of them moving as fast as possible, right? So you can't just keep increasing the concentration gradient and get it to increase and increase forever as you get more and more glucose outside the cell, because those proteins, there are only so many of them and they can only work so fast. So when we want to change the rate of facilitated diffusion, right, we're working with a baseline rate at which that carrier protein operates. The thing we can do is we can change the number of carriers in the membrane. Right? If you add more glucose transporters, you'll still eventually have it plateau out at based on that rate of transport for each of those carriers. It's going to be somewhere higher if you've got more of those carriers in the membrane in the first place. If we're at lower levels, so say our problem in getting glucose to get across wasn't that we have so much glucose that we're overflowing those carriers. Their problem was we only had like a little tiny concentration gradient. Right? You will still see an influence at those lower levels so long as you're below that maximum of increasing concentration gradient. Cool. So an example of this is something called glucose transporter 4. So glucose transporter 4 is just a name of a specific carrier protein that carries glucose. Okay? So you make glucose transporter 4 the way you make any other protein, right? So any other protein synthesis happens, and you make a protein by reading genes with ribosomes. In this case, particularly ribosomes embedded in your rough endoplasmic reticulum. Those transporters, any other proteins, get packaged into vesicles. They probably get sent through the Golgi body also somewhere along the way here for modification. But where we're picking up, where we're interested in, is the way that you get these GLUT4 proteins embedded into a membrane is that they respond to influence. Okay. So like, we're imagining a cell, right? So here's our cell. Make our transporter purple. Okay, so here's our membrane protein. Okay. So let's say we start out with just 2 of them at our baseline state. We've got more waiting in the wings in some vesicle inside, right? So a vesicle is basically just a bag of membrane, right? So a little membrane inside, storing something inside. Okay. So at our baseline state, we have a couple of these GLUT4 molecules, right? And I'll make our glucose, let's say red, right? So glucose can get carried across at this rate, right? It's only got 2 spots for it. All right. So when you eat food, when you eat carbohydrates, right, part of your goal to get those carbohydrates, get that glucose into your cells so you can break it down through cellular respiration, through all those metabolism steps, right, and get ATP. Right? So we're going to have, you ate some food, now we got a bunch of glucose hanging out. All right. So the reason insulin is important for your body, because now that you've got this huge concentration of glucose that you're starting to get into your bloodstream, right? If you still only have 2 transporters for glucose, that's not going to do you much good if you're trying to ramp up metabolism and make a lot of ATP, right? Because those 2 little glucose transporters can only work at their top speed. There's nothing more they can do. All these other little glucose molecules would have to kind of like wait their turn. Now, insulin does a neat thing. Insulin is going to take these GLUT4 molecules that are waiting in a vesicle. It's going to insert them in the membrane, right? So with insulin, we now have more transporters on the outside, which means now we can speed up our facilitated diffusion of glucose. So we're still working from high to low, right? Our direction is still high to low for glucose, so still down that chemical gradient with the chemical driving force. But now we can do it faster, even though it's passive. So we basically created more doors, more little buckets that are letting us get glucose into the cell. So that's fundamentally what insulin does. This is why your body releases insulin when, right, because it wants to be able to suck all that glucose that you're eating into the cell. In diabetes, one of the problems, right, if you don't make insulin, right, you're not going to be able to pull all that extra glucose into your cells. And now you're going to have extra glucose just like hanging in your blood, right? So that's why people's glucose levels would like spike if they are diabetic, why they would need to take their insulin, right? Because they're going to end up with just that overflow as these transporters aren't able to keep up with this increase in glucose concentration. Okay. But this is still all passive transport. This is just a type of passive transport happening spontaneously down, in this case, a chemical gradient. Right? We've just added a helper molecule in the form of this carrier. Diffusion through channels is similar, right? It's the same kind of idea. We've got a molecule, it's got a chemical gradient. In this case, when we talk about channels, usually we're thinking about ions, so we probably have an electrical driving force as well and an overall electrochemical driving force. But whatever, we still have a direction we want to go, right? We figured out the direction using those concepts. We've got a molecule or a particle that can't go directly between the phospholipids, so we need a protein cell. We're just using a different protein. So facilitated diffusion, we were thinking about carrier proteins. Diffusion through channels, we're thinking about channels, right? So these are still proteins. They're made of several different polypeptides, right? So you can see they have like multiple different little parts, right? But we're still just thinking about proteins that are going across the cell membrane. In the case of a channel, they've got a passageway through them. Okay. So here we've got like a hallway or a doorway going through the membrane. Okay. So in the case of a channel, right, it's going to be substance specific. So it's only going to let certain or specific molecules or ions or whatever through. But when it's open, we don't have to wait for this kind of like conformational change, right? We will have open and closed state, but once it's open, you can just have however many ions walk over. So there's a little bit of a consideration there that's different than when we're waiting for a carrier protein to change shape and kind of flip across. So still a transmembrane protein, just slightly different function to it. So types of channels that we'll see, actually, pretty much never draw them in on biology, except for when we are talking about acidity. But water actually moves through a channel called an aquaporin. We just have our baseline assumption that all your cells have aquaporins and all cells in general have aquaporins, unless we tell you otherwise. Okay? This is why you never really draw them on. So that'll come back in A&P2 because we have one specific case where it's actually important and some of your cells don't have them, but water goes through a channel. As we talk about neurons, channels are going to be super important because all of how your nerves work is going to depend on different types of channels that open and close at different times in response to different types of stimuli. Okay. So we're going to see different types of ion channels as we move into our next unit. So we're going to see leak channels, but as the name suggests, allow a little bit of leak across the membrane, but specifically of ions like usually we're thinking about potassium leak channels, maybe some sodium leak channels. We'll see that there are gated channels where there's like a wall in the middle of the channel and you need an event to happen for it to open, but then once it's open for a while. Sometimes we'll even have bidirectional channels where we can have things that flow in or out through the channel. It's just an open door, right? So we'll talk about all of those types of channels in our next unit and when we see them and what different that makes. Right now, we want to think about our factors affecting that rate of transport for each of those ion channels. So we don't have to wait around quite as long as we did for area proteins and facilitated diffusion when we're talking about channels, but that hallway still could get full, right? That door could be full. So there is a transport rate for channels as well. Number of channels in the membrane is going to make a difference, just the way it did in facilitated diffusion when we were thinking about carriers. You have more channels, you can get more stuff going across faster. And we're going to care whether they're open or closed. Right? If we have a closed channel, doesn't matter that the channel is there. If it's closed, nothing's getting across. So slow movement, we open it up, we'll speed it up. But in general, we're going to think about our rate for our carriers and our channel, so for facilitated diffusion versus movement through a channel is pretty similar. But really the difference in like the protein structure that we're going to be thinking about is a difference between those two types of passive transport. Okay. So questions about the three types of passive transport that we just discussed before we move to active.
Are like membrane like the carrier proteins that you wouldn't like the root or whatever you think about that. So would you have like
That's a great question. I don't know the answer to that. If there's a disadvantage of having them there all the time. I don't know. I can make up an answer for you. Like I wonder if they like decay, right? Or if they like go bad, but that is completely. You should Google it. Yeah. That's a great question. Yeah. Okay. Okay. So we're good on these types. Okay. Cool. I'm going to be thinking about that question. But we are going to move on for right now to think about active transport. Okay. So for active transport, you still want to start by identifying what direction your molecule or your ion wants to go relative to the membrane, whether it wants to go into the cell or out of the cell. Okay. But now we're going to be looking for a situation where we're observing that whatever this particle or molecule is, we're going to be noticing it's going the wrong direction, right? So if we have an electrochemical driving force going in and we see that ion going out, that's going to be a signal to us that this is active transport, right? Or if we have glucose and we see going from an area where there wasn't much glucose to an area where there's already a lot of glucose, that's a sign we're looking at active transport. So to have this uphill movement, right? So in the direction that is not towards equilibrium, in the direction that's harder, that's going to require energy. That's not going to happen spontaneously. And that is our definition of active transport, right? So what we're going to be looking for is this movement in the quote unquote wrong direction when we try and identify active transport. There are going to be two different types of active transport that we're looking at, but that's always going to be our key that we're looking for to tell that we're looking at active transport. So we see something going in the wrong direction. So active transport usually going to start with a pump, right? And anytime you see something described as a pump, you already know it's active transport. We don't use that word for passive transport ever, right? Pump implies you're having to put in energy to move something, right? Like if you have to pump something full of air, you have to move your arms, you have to put in energy, right? If you're running a pump across a membrane, same thing. Energy is going to be required. So the pumps look a little like those carriers, kind of, but there's a little more complexity. So what we're looking at in this picture is a picture of the sodium potassium pump, which drives like practically everything in your body as far as I can tell. Okay. So a pump is still a membrane protein. Like the carrier protein, a pump has binding sites, okay? Unlike the carrier protein, the specialized type of membrane protein also acts as an enzyme. Specifically, it acts as an enzyme that breaks down ATP, right? Okay. So it's going to act as both a transporter, right? It has the same like transporter carrier function, but it's also got a way to use energy, okay? So here we see specific binding sites here on our sodium potassium pump for sodium, right? So a pump has specific binding sites. In the case of our sodium potassium pump, that means 3 binding sites for sodium. Here we see that enzyme action in this intracellular portion. So your textbook likes to use blue for extracellular fluid, right? That water surrounding the cell. It's kind of peachy color for inside the cell, the intracellular fluid. So we can see this is taking energy in the form of ATP. It's breaking off that last phosphate group, releasing energy in that final bond. And that's the energy that we're going to use to flip the pump to the other side. Okay. So like our carriers that we were talking about with facilitated diffusion, we do have saturation so they can get full, right? And in fact, we are waiting for 3 sodiums to fill in these 3 binding sites. Now we will talk about the other side of that in a little bit when we have our potassium coming in on the opposite side, because sodium potassium pumps do a kind of like switcheroo. But first, I'm going to remind you that we have both primary active transport and secondary active transport as our 2 types of transport. And when we're looking at those pumps where we directly see ATP involved, that's our primary active transport. So that's like the first thing that needs to happen, I think is how they named it. But we can also just think of it as like our first type. You probably are easier type to identify of active transport. If we see ATP needs to be used as another way, we automatically know this is active transport. Okay. So let's get back to that pump. All right, so here we're looking at our sodium potassium pump. We can see that we have energy coming from that use of the ATP molecule, right? So we saw 3 sodium binding into these intracellular binding sites, right? So on the inside of the cell side. And we saw that breakdown of ATP, right? So we did this through a hydrolysis reaction, right? So requiring water. We broke, like this is always breaking something. We broke ATP. We got the energy from that bond to use, and it left us with this little phosphate group and our adenosine diphosphate, this lower energy ADP molecule. Okay. So once we break that, we use that energy to flip the sodium out to the outside of the cell here. So our sodium is now out in the extracellular fluid, increasing sodium concentration outside the cell. We'll see. That'll be important in a lot of our different cell types. Okay. But we're not done. We want to go back to our original state, right? So the other side of this pump, all right, next couple steps. So once we're flipped with our binding sites on the outside, this is when we're going to care about those other 2 binding sites here that are for potassium. So when we're flipped in this direction, opening up those binding sites for potassium, we're going to see 2 potassium ions coming in here, right? You can can see as they do this, that final phosphate group is actually kind of letting go, right? That one that we broke off from ATP. And as it lets go, that flips our pump back over. Potassium binding sites are now facing the inside of the cell. And this brings potassium into the cell, increasing potassium concentration inside the cell. So this is the full cycle for our sodium potassium pump. Okay. This is cool because it has an effect on concentrations of sodium and potassium. We'll also see that this does have an effect on electrochemical driving forces, kind of, because we are changing charges, right? We've just made the extracellular fluid extra positive, right? We put 3 sodiums out there, but we only traded for 2 potassiums coming in, right? So only 2. So we actually just made our cell a little more negative, right? Basically just took 1 positive ion away from the inside of the cell. Because when we look at that membrane potential, we're adding up all the charges inside the cell, all the charges outside the cell. And I'm sure I wrote this down for you. Okay. Cool. So primary active transport, main one to understand most critically is that sodium potassium pump. Anytime you see the sodium potassium pump, you should recognize it and know it is primary active transport. It's not the only type we'll see, but super, super important. It's usually the setup for secondary active transport. So in secondary active transport, you're not directly going to see ATP if I draw your picture, right? So this is happening second, right? You ran your sodium potassium pump. You set up usually sodium gradient we're looking at, okay? Now, secondary active transport, we're going to use that gradient that you just built up with lots of sodium outside the cell, only a little sodium inside the cell. And we're going to use it to drag some other molecule against its own gradient, right? So we basically stored energy in this example, a sodium gradient, right? So we used ATP to create a sodium gradient with lots of sodium outside the cell, only a little inside. Okay? And now we're going to use that sodium gradient. And sodium now really wants to get back inside the cell. We created a big concentration. And we made that inside of the cell kind of negative because we didn't replace all those sodium ions with cytosine potassium. Right, so its electrical driving force is actually also pointing to the cell. Right, so we've got 2 reasons sodium wants to get in. Okay, so sodium really wants to move in, and we're going to have some membrane proteins that move 2 substances at once. So in this case, we're looking at a membrane protein that lets sodium flow down its gradient. And anytime you see a green arrow in your textbook, it's telling you that that's the passive direction, that's the favorable direction. Okay, so sodium, if we just had sodium moving into the cell with the gradient, that would be the passive transport. We don't only have sodium moving. We've also got the glucose. So glucose, this arrow is red because we would be looking at a situation here where we're imagining we have high glucose inside the cell, right? So we have those 4 transporters. We've been pulling glucose into the cell, but we want to get all the glucose out of your blood in this situation, as an example. Right? So we have high glucose in the cell. We got low glucose out here outside of the cell, but we want to get all the glucose, right? You ate that food, you want to get all the energy, right? So the secondary active transport is using how much sodium really wants to come back into the cell, and it's grabbing glucose and pulling it with it, basically, right? So we have this one protein that allows sodium to move down its gradient, but the second part of the action of that protein is to move glucose against its gradient, so in that active direction, in the harder direction. So it's secondary active transport because we're like using a second molecule as our energy source. So we're using a gradient that we built up already through primary active transport as our energy source. So there's still energy being used here. It's just not energy directly in the form of ATP. This is like a second step later on. So you can still recognize that this is active transport by making sure that you're identifying that at least one of these is going up its gradient. Okay? So you will always expect to see in secondary active transport that one of your molecules or ions or whatever is going in the direction it wants to go. The other thing is going in the direction it doesn't want to go. That's key. That is what happens in secondary active transport. So this was our example of a sodium-linked glucose pump. So a different way that we get glucose inside cells. Right? If we have a gradient of glucose where we have high glucose outside the cell, low inside the cell, all we need is facilitated diffusion. Let glucose go the direction it wants to go through one of those carrier proteins. But if we want to suck up all the glucose out of our food, we're going to need to actually power that process of getting the last little glucose molecules in. So we might do it by running the sodium potassium pump, creating a sodium gradient, and then using that sodium gradient to power our sodium-linked glucose pump. Questions about these two types of transport, primary active and secondary? Okay. Cool. I don't want to give you the impression that our molecules are always going to go in the same direction. So sodium and glucose were both going into the cell in that previous example, right? So they were like sodium was dragging glucose along. But we can also have secondary active transport examples that look more like kind of like a revolving door, right? Like at the front of, I don't know, say hospital, right? If you have someone running behind you really fast, it might push you through the opposite direction. We have transporters that work like that too. So here we're looking at a sodium-linked proton pump. So protons, hydrogen ions are basically the same thing, right? So we still have sodium moving down a gradient, right? So we have sodium going from, I'll use that. Okay, sodium going from this high area of sodium to this area of low sodium. That's our concentration. And we happen to actually know that most cells have a membrane potential of -70. So we also have an electrical driving force for sodium going into the cell. So this is part of why sodium is so useful as a way to power secondary active transport, because it's like double strong desire to go into the cell. Okay. So in this case, we've got that diffusion of sodium in the direction it wants to go into the cell. And this transporter, this sodium-linked proton pump, as it does that, along the way, it has to pick up a hydrogen ion or proton and push it out of the cell to penetrate. That's the way that this specific protein works. This is the way a sodium-linked proton pump works. But it's still using that sodium gradient, that sodium movement as its energy source. So this is pumping against that hydrogen ion gradient. So if you looked at the hydrogen ion gradient, you'd see it has a really high concentration here and a low concentration here. And we know that must be true, and that must be a really strong gradient. And we know that those hydrogen ions would otherwise be attracted to that negative membrane potential and kind of want to come in, right? So we're fighting against a lot of forces for these hydrogen ions. Okay. So this is active transport because we can see those hydrogen ions going the direction they don't want to go. Okay. So here's a big chart from your book about all the different transport processes. So we've got our simple diffusion as our first type of passive transport. We've got our channel movement, our facilitated diffusion as our two other types of passive transport. And we have our two types of active transport. And this is telling you what direction you'd be looking for to define each of these, what type of protein you're looking for, whether you need energy, whether those methods of getting through the membrane can get saturated, right? So you're not going to have to worry about saturation if you're talking about simple diffusion, because the membrane is the membrane. But you can see that for our active transport pumps, definitely there's saturation. Facilitated diffusion through that carrier protein, yes, for sure, saturation happens. channels are like harder to block, but could theoretically sometimes happen. So we got sometimes there, kind of like wiggly one. Here we also are showing that all of the times that we use proteins to move things across cells, it's going to be specific to a specific ion, a specific molecule. And here we can see what types of molecules are going to be moving by this process and some examples of ones that we'll see later on. So here's a big picture of how this all works together. Okay. So we're going to see the reason we have different concentrations of stuff in the intracellular fluid, the ICS, versus the extracellular fluid is because we have transport happening. Right? If we only had passive transport, everything would eventually reach equilibrium, right? Like it would all just move until you have equal amounts on both sides, movement would stop, net flux would stop. So because we have active transport, we're actively using energy to move stuff, particularly sodium and potassium. This means that we have differing concentrations of all sorts of things across the two sides of the membrane. Cool. All right. So we are going to stop here. There will be, as usual, a quiz up on right things for you to do over the weekend. I'll get that up momentarily. Sorry, my Thursday was a little weird, but that's coming soon.